molecules. The B cell produces only one kind of antibody, specific to that particular antigen. The antibodies bind with the antigen, which facilitates their disposal by the body. When the antibody production is no longer required, another type of regulatory T cell, called a suppressor, switches off the B cell. So each B cell is part of a trio with a helper and a suppressor T cell, all responding to the same specific antigen. Yet another type of T cell, the cytotoxic T cell, has the ability to recognize specific antigen on the surface of both bacteria and infected body cells and destroy them directly. Embedded in all cell membranes are numerous proteins, carbohydrates and glycoproteins, some of which serve as labels or marker molecules. These marker molecules identify the type of cell this is, a macrophage or a liver cell or a B cell perhaps. Other molecules identify the species from which the cell comes. And there's also a set of molecules that are unique to the individual of the species. And these are called the major histocompatibility complex, or MHC. And some of these MHC molecules appear on every cell in the body. Now, the MHC is as unique to an individual as a fingerprint. For example, it's certain that my MHC molecules are different from yours. And unless you have an identical twin, it's equally certain that your MHC molecules are different from those on the cells of every other living creature. Now, it turns out that the MHC molecules have a crucial role in the interaction of T cells with antigens, and we'll be coming back to that later. Now, all these surface molecules don't simply act as passive identification labels. They're also receptors, which can convey information into or out of the cell. So the obvious question is, how do they do that? How do they interact with other cells and molecules in the body? Well, more than 20 years ago, it was discovered that B cells interact with antigens by carrying embedded in their surface membrane a molecule of antibody with the combining sites for antigens facing outwards. Antibodies are probably the best understood molecules in the immune system. With a molecular mass of 150,000, they are even large enough to be seen by an electron microscope. They appear to have approximately the shape of a letter Y. The backbone skeleton of the molecule bears out this general impression. The overall structure appears complicated, particularly when all atoms are displayed in a space-filling model, but both chemical and structural studies have revealed an underlying pattern. They show that the molecule displays a simple regularity. It is composed of 4 polypeptide chains connected by disulfide bonds. The long chains are known as heavy chains, and the short ones as light chains. Both are made up of repeating units or domains. In the case of immunoglobulin G, or IgG, the most abundant class of antibody, there are 2 in each light chain and 4 in each heavy chain. The domains show a great deal of similarity, both in amino acid sequence and in 3-dimensional structure. Those which do not vary from molecule to molecule are called constant domains and make up the main part of the antibody. The others, which make up the tips of the Y, vary slightly for each specificity and are called variable domains. It is these variable domains, one from the light chain and one from the heavy, which contain the binding site for the antigen. They are folded into 2 grooved barrels, which lie side by side and form the framework of the binding site itself. The sides of the barrels are made up of strands of regular sheet structure with interconnecting loops. The inner walls of the light and heavy chains, shown here in red, make up the sides of the combining site cavity. Some of the loops which protrude from these beta sheet walls vary greatly in sequence and sometimes in shape from one antibody specificity to another. It is these hypervariable loops, 3 from the heavy chain and 3 from the light, which determine the shape and chemical nature of the binding site, and therefore the type of antigen it recognizes. It's easy to see what an economical and yet adaptable structure this is, entirely built from repeating constant subunits, with the variability necessary for antigen recognition confined to a relatively small part of the molecule. Now, the genes underlying this structure turned out to be far more interesting than anyone could have predicted. The genes that code for the constant domains are common to all B cells, but the variable domains have to be produced in more than 100 million different conformations in order to combine with all the antigens that we know exist. Now, there simply isn't enough DNA in the cell to have a single gene for each one of these different conformations. The diversity is achieved by rearranging a relatively small pool of genes. Originally, the DNA contains multiple alternative genes coding for the variable domains of the antibody molecule. The DNA coding for the antibody light chain contains about 300 different variable or V genes, each capable of coding for the variable regions, plus a few joining or J genes which also contribute to the structural variation. Further downstream come the constant domain or C genes. Early in development, each B cell selects a V gene at random, and the DNA which lies between the chosen V gene and the J genes is excised by enzymes and the DNA strand rejoined. The DNA sequence from the chosen V gene to the C gene is transcribed into messenger RNA. Next, enzymes trim the RNA even further by excising all but one of the sequences transcribed from the J genes. This leaves a unique combination of V and J gene transcripts. Finally, the messenger RNA is translated into a protein light chain with a unique variable domain. A similar process of gene rearrangement occurs in the heavy chain. The idea that genes could be rearranged to generate diversity within antibody molecules was a revolutionary one. It was first proposed by Susumu Tonegawa, now at the Massachusetts Institute of Technology. He recalls the conceptual shifts that had to be made before his ideas could be accepted.
Many biologists thought that genes are very static in the lifespan of individual. So the idea that the genes rearranges during the B cell development was against that dogma. That's one. The second is there is this another dogma in molecular biology or genetics, which says one gene encodes for one polypeptide chain. Okay? So the idea of the variable region and the constant region encoded by separate genes sounds like there are two genes for one polypeptide chain. So that was the second point, which were not necessarily accepted by mainstream of biologists. So our finding went against both of those. That's a conceptual difficulty. But once it was shown, since the experiment was so direct, experiment data was so direct, it was quickly accepted by many.
When the genetics underlying antibody diversity was finally unraveled, it seemed reasonable to expect that the other family of lymphocytes, the T cells, would also recognize antigen by carrying an antibody-like molecule in their surface membrane. But despite years of exhaustive research, no such molecule was found. Whatever the T cell receptor for antigen is like, one problem in studying it is that T cells, unlike B cells, don't secrete large amounts of their receptor, which can be isolated and purified. Another problem is that T cells recognize antigen in a much more complex way than B cells. T cells can't recognize free antigen, but only antigen that's presented to them on the surface of another cell. Moreover, it has to be an association with the major histocompatibility complex, the MHC molecules. The question of how T cells see antigen plus MHC is still not resolved, but a major breakthrough in immunological techniques produced rapid progress. It became possible to induce individual T cells or B cells to divide continuously in tissue culture, each one giving a clone of identical daughter cells. Now, if a B cell is cloned, all its daughters secrete large amounts of identical antibodies, all binding with the same antigen. These are the so-called monoclonal antibodies, which have revolutionized many areas of biochemistry. Now, T-cell clones can also be made, and similarly, each member of the clone carries an identical surface receptor for antigen. Once identical T cells were available in large quantities, it became possible to start work on trying to characterize the structure of the T cell receptor. At the Dana-Farber Institute at Harvard Medical School, a group of biochemists and immunologists began to use these new techniques to study the T cell receptor. Just as Susumu Tanegawa had to overcome the accepted dogmas in unraveling the antibody structure, so Ellis Reinherz, the leader of this group, faced a confusing array of theories about the nature of the T cell receptor.
The current thinking at that time was rather confused. Some people were of the opinion that the T cell receptor had already been defined. Some people felt that it was related to immunoglobulin. Others felt that it was related to a secreted set of structures which had been defined in one type of T lymphocyte. The sizes and molecular characteristics of molecules which were putative T cell receptors really went across the board. There was no general consensus. Our assumption really implicitly was simply that a T cell receptor which is responsible for the unique specificity of a T cell had to be expressed at the level of the cell surface. And so our goal was to produce antibodies which would react uniquely with a cell surface constituent on a T cell of a particular specificity and not on a T cell of a different specificity.
Reinherz's group generated a number of different T cell clones and used them to raise unique monoclonal antibodies, each of which blocked the ability of a single clone to recognize antigen plus the self-MHC molecules. The antibody's ability to block antigen recognition indicated that it must be bound to the T cell receptor. Later, these antibodies were used to identify and to isolate the T cell receptors from cell membrane fragments. With painstaking biochemical analysis of isolated receptors, they were gradually able to work out the amino acid sequence of part of the molecule. When receptors from different clones of T cells were broken down into peptide fragments and compared, it became clear that certain peptides varied from one clone to another, whereas others were common to all clones. This suggested that, just like the antibodies, T cell receptors were composed of variable and constant regions. In fact, the T cell is composed of two polypeptide chains, alpha and beta, each with a constant and a variable domain similar to those of antibodies. It looks like one arm of an antibody molecule. Although they didn't know at the time, all the major groups were working on the beta chain. Reinherz's group had discovered that monoclonal antibodies could be used to block T cell receptors, but could they also be used to switch them on?
We were able to switch on T cells by a rather simple maneuver. In this case, we used T cells which were derived from myself, since I have an enormous hay fever allergy during the fall months, and it's easy to generate cells from myself which are specific for the ragweed E antigen in the context of one of my own MHC molecules.
What they did was to raise monoclonal antibodies to these cells and bind them to beads. When the fixed antibodies were mixed with the T cells, instead of blocking them, they actually triggered the cells. What seemed to have happened was that once the antibodies were anchored, adjacent receptors on the T cell surface were sometimes connected by an antibody bridge, and it was this cross-linking that mimicked the combined effect of antigen and MHC together and triggered the cell.
It became imperative to demonstrate that this was in fact the amino acid sequence of the very beta chain which we had been analyzing.
Knowing this sequence, modern techniques made it possible to synthesize a peptide, which, if their assumptions were correct, would be identical to the original peptide in the receptor. The proof that it was indeed the same came when they showed that antibodies raised against the synthetic peptide also blocked the T-cell receptor.
This, in essence, closed the loop and indicated clearly that one had sequenced the beta subunit as opposed to something else.
While the work was continuing on the protein structure of the receptor, other groups were looking at the DNA within the T-cell. At the University of Toronto, the molecular biologist Tak Mak was using a technique which probed for genes unique to T-cells.
The technique that we used is what is called subtractive hybridization. It's based on the assumption that if you were to clone some genes that are in one cell and not found in the other, that what you do is would try to subtract away by hybridization everything that is common. And then what's left over then would be everything that is different. And you hope that it's not going to be so many genes. And so what you're going to do is to search among these whatever number of genes. It turned out to be around 20 or so different types of genes that are unique to T cells. You search among those 20 or so, which one may have the property that you would expect for a T-cell receptor.
Other groups were also in the race to publish their research on T-cell receptor genes. While Tak Mak was working on a human gene, another group, led by Mark Davis, was doing similar work on a mouse gene. But were they both the T-cell receptor gene?
We were each aware of each other's presence and that each other has cloned what we both thought were the T-cell receptor genes. So we tried to communicate over the phone and to try to determine if we have cloned the same gene, because at that time, nobody knows what the gene looks like. But however, after several attempts, we couldn't get to agree as to whether he should send me his sequence or whether I should send him his sequence. So as a result, we decided that we'll agree to disagree and then wait for the paper to come out. The whole revelation came out within 5 hours on the way from Los Angeles to Salt Lake City on the plane in March of 1984, where all the immunologists was congregating to attend this meeting. Somebody handed me a copy and said, "It's the same gene," and I was really rather relieved. Then a few hours later, when we finally got into Salt Lake City, Dr. Benacerraf walked up to me and he said that it looks like that after he had seen the data, that our sequence matched those of Alice Reinhardt's with the protein data for beta chain, and therefore the gene that we cloned was the same as Mark Davis' gene, which was the same as the beta chain, identified by the basic protein that was in the immunoprecipitation of using the monoclonal antibodies.
When Mak had isolated genes which he believed coded for the T-cell receptor, he devised an experiment which provided direct confirmation.
The method we used to prove that the gene that we've cloned are actually the T-cell receptor genes was to use gene transfer experiments. I gather that after the alpha and the beta chain genes were cloned, that they had the properties that one would expect of a T-cell receptor, in that they undergo somatic mutation to generate diversity, they have immunoglobulin-like structures, but the final proof that they are indeed the T-cell receptor genes have to come from a gene transfer experiment. And what Pamela Ohashi, a graduate student in my lab, did was to clone out the genes of alpha and beta chain, the T-cell receptor, and then to use gene transfer technique to transfect, or to introduce these two genes into a cell which is incapable of recognizing a particular antigen. And by introducing purely two pieces of DNA into a completely foreign cell, this cell now acquires the ability to recognize the antigen. Then that in itself conclusively proves that these genes encode for the information necessary to recognize the antigen, and therefore it is the T-cell receptor.
The discovery of the nature of the T-cell receptor is a striking example of the combination of protein biochemistry and molecular biology. Although many questions remain unanswered, this research has achieved a major advance in understanding immune recognition. We asked Tak Mak what consequences there might be for clinical medicine.
I would speculate, and there is already data coming out from our laboratory in collaboration with two Japanese groups, that at least in mice, the T-cell receptor genes are directly related to autoimmunity. And therefore it is going to be one of the missing links. And the work will have to go on to prove that relationship exists in some type of autoimmunity in humans, and I'm confident that that will come.
In the next program, the course on